Welcome to the Performance Around the Clock podcast. I am Professor Satchin Panda, a scientist in the field of circadian rhythm. In this podcast, I will talk to experts in many different fields about science, health, and human performance. You'll also find out why circadian rhythms is my favorite topic. I hope you enjoy the podcast and thank you for listening. Welcome to another episode of Performance Around the Clock. And this is from San Juan, Puerto Rico, where we are having this circadian, the largest international circadian conference of the year. And I'm super excited to be sitting down with Professor Achim Kramer from Berlin. And he is considered now the leading expert in someone who is always thinking, sleeping, dreaming about circadian medicine, how we can bring the knowledge, the wisdom of circadian rhythm into preventing, managing, and curing patients suffering from many different types of diseases. Welcome, Ahim.
Thank you, Satchin. Happy to be here.
Yeah. So, we all in circadian rhythm field have a very different way we got into the field. The thing is, people go to school or college and then they say one day they will be a doctor or one day they will cure cancer. But did you think about becoming a circadian rhythm researcher?
No, at the beginning, I wanted to study mechanisms of life, and I was trained as a biochemist, and I did my PhD in structural biology and also my first postdoc. And the funny thing, how I came into circadian rhythms was that I was reading in a regular Berlin newspaper about an article published by someone at Harvard Medical School about discovery of a clock gene. And I didn't even know at the time that something like a circadian clock exists, and was just emailing to Clark Weitz at Harvard Medical School, and I ended up doing my second postdoc with him.
Wow.
And that's how I came into the field, and I'm excited about it.
Yeah. No, that was an exciting time. And Chuck does I think he did some of the best biochemistry of circadian clock mechanisms. And so then how did you see what are the next phases of circadian rhythm research?
Yeah, I mean, when I came back to Berlin and had my own lab in 2002, the first 10 years or so, I was kind of working on really the cogwheels and the phosphorylation of clock proteins, everything which made the molecular mechanism. And then at some point, we also had a little bit work on immunology and the clock. And then this kind of translated into looking at maybe we can use that molecular insight into the clock to create, for example, a diagnostics in humans, because I'm at the medical department at Charité. This is the biggest university hospital in Europe, and so also my colleagues were kind of asking me, what are you doing and how can we do that translated into the clinic? And this is how the second 10 years after I founded my lab kind of were dominated by this.
I didn't know that Charité is the largest hospital in Europe, university hospital. So whatever you do there will actually have a huge impact.
In the last, I think, 5 or 10 years, I have built a network with many clinical colleagues, trying to first of all educate them about circadian rhythms, and then to convince them, and that was easier after the Nobel Prize, that circadian rhythms are important for health, and disrupting circadian clock is a risk for your health, and you can use that in the clinic for the benefit of the patients. And this is why the environment in a hospital is good for doing this.
Well, I think that you alluded to this point that you got into circadian rhythm and immunology. That's the macrophage work, right?
Yes.
Yeah. So right now it's becoming more and more clear that immunity or immune dysfunction is the driver of many chronic diseases, starting from diabetes all the way to cancer and Alzheimer's. But what was the thinking? How did you get into circadian rhythm, biochemistry to immunity and macrophages?
I was also a little bit of a coincidence because I ended up when they hired me, and there was an open topic call and they didn't know where to put chronobiology, and I ended up in the medical immunology department. And this I had many immunologists around me, and therefore we were kind of motivated to also look at immune cells. And I think our 2009 paper about circadian clocks in macrophages was one of the looking at immune system, and that was also important for our later work in looking how can we use these rhythms for diagnosing, because immune cells are easily accessible cells in humans, minimally invasive. You can draw blood, and we knew from our mouse work with macrophages that macrophages / monocytes have one of the best clocks, and this is why we focused on those cells in blood to use them for predicting phase in humans.
I see. And then macrophages are also the cells that go eat stuff. That's why they're called macrophages, right?
Exactly.
So they are present in almost every organ.
Yeah, they're present in almost every organ. They kind of travel between bloodstream and organs. In the liver, they're called Kupfer cells. In other organs, they're called differently, and they are presenting foreign antigens. They're presenting cancer, peptides, and so on. So they're really important for our, when we call it, innate immune system.
Yeah. No, you said that inspired you to look for circadian diagnostics. So that means figuring out whether your circadian clock is normal, abnormal, etc. What is the state of the art right now for a consumer?
For a consumer, you can buy a test
outside your test. Like what I'm going back to before your test.
Okay, people may know what kind of type they are in terms of sleeping type, whether they're an early type or a late type or something in between. They have a feeling, but often they cannot live according to that feeling because maybe they have little kids, and maybe the school starts too early, and there is, like in height, there's a almost normal distribution of many people being something in between, and a few late and a few early. And it was always kind of a subjective feeling of what you are, and there are questionnaires which are good, which you can fill out and get an answer. You are an early type or a late type, but there was never an objective measure until such tests were, I should say, there was objective measures, but not easily accessible.
Like the melatonin?
Yeah, the melatonin assay could have been done. It has not been used in the clinics, has not been used for the consumers, but in research, people have measured the time when the dark hormone melatonin is secreted by the pineal gland, and when it reaches a certain threshold, this is considered the gold standard of chronotype, circadian phase.
But it involves taking sample in every 1 hour, 30 minutes, or something like that for 4 or 5 hours.
It's super impractical, usually 6 hours, and you need to sit in the darkness, in almost darkness, because light suppresses this melatonin secretion, and you need to kind of do it outside of usual doctor's office hours. So it needs to be in the evening, because that's the time when it goes up. So it was impractical, and it's also expensive.
Yeah, so there are 3 things. One is you're doing the test outside the doctor's office hours, because your melatonin will go up only before your habitual bedtime. So you have to do it either at home which is also difficult because somebody has to collect samples, or go to a specialized clinic. And then you draw blood or saliva in some cases, I think, in every 30 minutes or 1 hour, sitting in a dark room or with a little red light, and you cannot have actually watch TV or anything, right? Because that
Right. Yeah, so it's super. So this is not a scalable thing.
No, and that's why I think there is no, no doctor ever prescribes a melatonin dim light melatonin onset acid. I don't think there is even a code for that.
Exactly. I mean, even
I may be
in
wrong.
ICD-10, so this is the diagnostics catalog.
Yeah.
If there is a circadian rhythms and sleep disorder, and there are also these advanced sleep phase disorders and delayed sleep phase disorders. The delayed sleep phase disorders means you cannot sleep at times when you want to sleep. You just cannot fall asleep.
Yeah.
And this is a disorder if people suffer from it. And in this diagnostic criterion, this is not even a part of it.
So melatonin is not part of the diagnostic.
It is mentioned as supportive, but it's not part of the diagnostics.
I see.
Yeah.
So right now, in most of the world, if people want to know what is their circadian rhythm, because people always ask me, hey, how do I know what is my circadian rhythm is? And then they say whether they're early chronotype, late chronotype, but as you say, it's very subjective. And also it's culturally subjective. For example, I come from India, and in India, waking up at 6 o'clock or 5:30 a.m. is considered normal. Whereas in many parts, it can be early. So
yeah, I mean, this is interesting because we should be clear that chronotype is not something, or it turns out more and more, it's not a trait so it's not static.
yeah.
It depends on many things. It depends, what we know from questionnaire data, it depends on age, it depends on sex, which is not changeable, but you age. It depends on the amount of light you get.
Yeah.
And Till Renneberg has investigated the chronotype distribution in Western Europe and compared it to India. And because you mentioned India, and people in India usually get more light.
Yeah.
So, and that makes them earlier.
Yeah.
And that makes them the distribution narrower, so you don't have so many extremes.
Yeah.
And it also, so it depends a lot on light, and it turns out it may also depend on season.
Yeah.
Right, because especially in countries where you have a lot of season, so it's not something where you think you are this chronotype with this exact time and ideal bedtime. It depends on the environment, but also mainly on your genetics. So it's a combination of nature and environment and nurture. Exactly.
So when we talk about in circadian rhythm field, there are many technical terms.
Yeah.
And two terms that we always use is phase and amplitude.
Yeah.
Can you expand on these two terms and why it is relevant for everybody to know a little bit about what is phase and what is amplitude?
So phase is actually when things happen.
Yeah.
So if you talk about phase, then it may relate to when is the best time to take your medicine, what is the best eating window, that's phase, and what is my status in terms of am I internally, is it in my body, is it night or is it already day? And if you are a school kid or a teenager and you go to school at 7 o'clock or at 8 o'clock, it may be internally an earlier phase.
Yeah.
So you are still probably in the middle of your biological night. So that's phase, when things happen.
Yeah. So what time it is inside my body?
Yes, exactly.
Right? Yeah.
And for the scientists among us, we call that phase of entrainment, because we are living in a 24-hour light dark cycle, and with relation to that, what happens in my body. So if for example, we talked about melatonin, if my melatonin rise in the evening is late, then this is a late phase of entrainment.
Yeah.
And amplitude is
So
based,
let's go back to the melatonin stuff, because that's something that some people may relate to. So for example, what you're discussing is, suppose the sun sets at 6 p.m. Hypothetical, of course, the sun will set at
a little bit like here, in Puerto Rico.
In Puerto Rico, sun sets at 6 p.m. And then if we do this melatonin, dim light melatonin assay, so for example, my melatonin may begin to rise at 9 p.m. So that's my internal time of melatonin onset. If we say melatonin onset tells me what time it is for me to go to bed, for example, then that's 9 p.m. And for you, suppose say it's 11. So I'm just making up.
Yeah, you're making it up. I'm a little earlier.
Suppose I am 11 and you are 9. Okay, let's do. So mine wakes up at 11 and yours is at 9. So this is an important information, because for you might say, well, I know my internal clock at 9 o'clock is telling me I should sleep. Whereas for me, at 9 o'clock, my internal clock is telling me, no, you can still stay awake for 2 hours. So I think that's the phase. And if we know what our internal time is telling us, then we can decide like when to go to bed or when to wake up. So for example, if you're going to bed at 9 and if you want to sleep for 8 or 9 hours, then you know what you're...
And it does not only refer to sleeping. It refers to eating. It refers to when is the best time to exercise, what type of exercise, when is the best time to take medication, and so on. I mean, the phase is really important because it tells you when to do things and when to do it in the best alignment with your internal clock.
So for example, when to eat. Again, now we know a little bit interaction between melatonin and food and glucose tolerance. So that means for you, sorry, Achim, your last meal should be around 6 or 7 p.m. So there is no, the good thing is you don't have to spend too much on drinks. But for me, if I stop 2 to 3 hours before, I can still go.
So I have to buy you the drink, and you...
and then I can keep drinking and do the practice. So these are important points that you brought up. This important point, that phase. If we know what time it is inside our individual body, then we can figure out what is the optimum time for, as you said, exercise, food, and everything.
Exactly.
And then what is amplitude?
Amplitude is a bit more complicated.
Yeah, it is.
But at first sight it may be easy because it just tells you how strong your rhythm is. So how big is the difference between, for example, the maximal melatonin secretion and the minimal melatonin secretion, or the maximal stress hormone secretion, and so on. And but why is it sometimes lower and sometimes higher for people? And this can be, it's not super clear because it can happen on several levels. So at first glance is maybe one level might be your internal clocks, because we have them in every cell of our body and not only in the brain, maybe not perfectly synchronized. So they run at different, now we are at the term phases again, they run at different phases, and globally kind of adding up all those phases makes the rhythm weaker, because it's like runners which are not kind of tied together with when you have several cycles in a running, then they kind of spread out, and then overall the rhythm gets lower. But it could also be a second possibility is that the individual clock rhythm has a low amplitude, and that would kind of be visible in every cell. And it's not necessarily a matter of how synchronized the cells are, but it could be that the individual cells have a low amplitude. This is to be characterized better in the future.
When you're talking about these cyclists starting biking around it, I was visualizing the stadium, and then, in the stadium, when people do those waves, they stand up.
Yeah.
When they stand up in sync, it looks beautiful. It's very nice rhythm. But then when people stand up out of sync with each other, they may be standing up, but then it doesn't.
And the more out of sync they are, the less the amplitude is. And we can then see this is a kind of good a metaphor for amplitude.
Yeah. So for example, nowadays there are a lot of watches that can sense temperature. Although the temperature is very fluctuating, because depending on what we may be doing, our skin temperature.
Yeah, you mean the body temperature.
Body temperature. If you are going outdoor in Puerto Rico in the middle of the day, it can be pretty hot, and your skin temperature may give you a different reading. But set aside, suppose that somebody is most of the time staying indoor with air conditioning and also nearly constant. Still, the body temperature would cycle.
Yeah.
Is that a good indicator of amplitude?
Actually, when I talk to colleagues and ask them, do we have something like a gold standard for circadian amplitude, they will also always say no. And which is different from phase, because from phase we talked about that this melatonin rise in the evening, this is considered the gold standard. But we don't have a good gold standard for amplitude. We have been looking at data where we have people in a so-called constant routine protocol where we have like body temperature rhythms, melatonin rhythms, cortisol rhythms, gene expression rhythms, and try to see whether these amplitudes correlate, and this is not so easy. So we are still have to define, or we still have to work on how to best measure circadian amplitude. But I could imagine that the skin temperature may be a first proxy, which may be helpful.
But again, it will be a very narrow range, right? Because our body temperature, if we said body temperature, that's usually core body temperature. Skin temperature is inversely related. It also has a very narrow range. Our temperature doesn't fluctuate.
Maybe at 1.2 degrees max.
1.2 degree max. And then
or even less.
You can have a ceiling effect.
Yeah, you can have a ceiling effect, and you have, as you mentioned, you have a lot of, and even in an environmentally relatively constant condition, you still need to move, and every time you eat, your body temperature goes up. And this will also, and we call it masking, and can mask the internal circadian rhythm amplitude. So maybe we need something else to measure amplitude.
The bottom line is we don't have anything now.
Yes, true.
What about blood pressure, for example? How much does it changes 4 or 5 points or something like that?
I'm not so sure. I know. I know. If you go to the doctor and the doctor measures your blood pressure, and you do it in the morning, it is higher than in the evening, and this may be influencing on whether the doctor prescribes you a hypertensive medication or not. So the doctor should know that. I mean, there are maybe the future is that you measure multiple of those and maybe we at some point have a device which measures blood pressure, which measures temperature, which maybe measures cortisol in interstitial fluid, which or automatically, and then it measures a little bit of glucose, and all these together, and together with light and movement and an intelligent AI-based algorithm, we may be able to extract amplitude.
No, recently I'd seen a couple of papers where they're integrating activity from wearables, and then I think glucose monitor, and then also sleep or light exposure. But you're right, means your example of different rhythms being out of sync can also reduce amplitude. You can imagine, for example, blood pressure not going down at night.
The non-dippers.
They may stay constant, but the temperature might still oscillate. and the sleep-wake cycle may be out of sync. So that can be a sign of. Now we'll come to the next topic. What is circadian disruption? Because
Exactly.
we use that term
Yeah,
a
the
lot.
circadian disruption is probably a good term, but it's an umbrella term for many different things, in my opinion.
I think there are quite a few similarities. There are a few terms we biologists and even in general we use which are difficult to define. One is circadian disruption, and in immunology, inflammation.
Inflammation. Inflammation, yes.
Yeah, it's very difficult to define what is inflammation.
But since you mention it, I think it's very interesting because I think these two things go hand in hand.
Yeah.
So you have when you're circadianly disrupted, then you are probably as in obesity, or so you have a pro-inflammatory state, and this is probably correlated with circadian disruption. But what is it? It could be maybe I throw out a couple other of terms, which is circadian desynchronization, circadian misalignment, circadian so that also means that maybe the most generic definition is that the temporal coordination is not normal anymore.
Yeah.
So and that could be on several.
But it's very difficult to define what is normal.
Yeah, it's very difficult to define what is normal. But you can, I mean, yeah.
I mean, let's take, we can try to, like, for example, melatonin rhythm in ideal environment where we can actually measure melatonin, which is not possible, of course, for everybody. I think what we can say is melatonin should begin to rise around bedtime, plus or minus 2 hours or 3 hours.
It's usually a little bit earlier than bedtime. Yes.
Yeah, so it's 2 to 3 hours before.
Yeah.
And then it should peak and go down 3 to 4 hours before we wake up. And then
at that time, usually you have high cortisol.
Yeah, your cortisol should be high.
Yeah.
And usually cortisol should peak within 30 minutes of waking up, 45 minutes of waking up. And that's when melatonin should be still going down. So that we can, I think many human experiments have shown that may be qualitatively normal, but we cannot say what should be the melatonin level, what should be the cortisol level.
I mean, when we look at gene expression in easily accessible tissues such as hair follicles or monocytes or blood cells, then we see, of course, we also see nice rhythms, and we can define the amplitude of those rhythms, and we can define the phase relation of those rhythms, because there's certain genes which peak in the morning, other gene activities are maximal in the evening, and so on. And when this pattern of these different phases of the different genes is looking like it should, and looking like as we know it from animal models, then we can see everything is kind of normal.
I see. Yeah. So you can actually define a molecular normal.
Exactly. We can define a molecular normality in when we assume that we also are kind of mice.
Yeah.
Right.
Yeah,
so because for mice we can do very standardized conditions. Every mouse is genetically identical, has the same age. We have the same light-dark cycles. They get enough water, enough food, and this is a perfectly standardized normal condition. And from those experiments, we know the genes and the gene activity and the phase relation of these genes. And if we find this in humans, then we can define this as normal.
So if we go back to your experiment, the macrophage gene expression data set, in humans and mice, we share many of the genes. There are nearly a dozen or so what we call clock components, and they do cycle at different time. They peak at 2 or 3 different times of the day. So are you seeing those rhythms? So we are talking about those, instead of looking at hundreds of genes cycling, can we just look at those dozen or so?
Yes, we can. I mean, we have asked the computer, or I should say maybe artificial intelligence to extract for us out of the 10,000 genes or 20,000 genes or maybe 1,000 oscillating genes, those which are the best predictors for a typical time of day. Because you can imagine, if I take a blood sample at 9 o'clock, what I expect to see is the 9 o'clock genes most active, and then the 9 o'clock in the evening genes least active, and the midday genes a little bit, and so on. And if I, for example, take a 9 o'clock sample of a late chronotype, then what I see then is that the night genes are still up, and the morning genes are not yet up. So I know in this guy it's night inside, even though I took the sample from 9 o'clock. And the challenge or the difference between mice and humans here is that the computer or the artificial intelligence not only extracts genes which have a nice rhythm, but also genes which are most similar between you and me.
I see.
Right? Because it doesn't, because sometimes circadian genes, which we know as clock components, oscillate in you and oscillate in me, but on a different magnitude level. So that would not be good predictors, because the peak of my expression might be the trough of your expression. So we have
because
two
your waves
challenges.
may be pretty high, and my wave may be low.
Exactly. Even though we would have the same phase, and we would even have the same amplitude, but we would not have the same expression level. And if you want to kind of use a single sample to predict phase, you need to rely on the same level.
So what you're saying is, for rest of us, all the clock genes, let's go back to this 12 or some genes that you figured out are most predictive. Those 12 genes or so, they don't actually reach their peak at the same time, right?
Not necessarily.
Not necessarily.
Because if we are 2 hours apart, then it reaches for me earlier and for you later.
No, so even those 12 genes. So for example, so this is very important for everybody to understand, that those 12 genes don't have to be in the same. They actually don't, in general, in normal condition. They're not picking all of them at 9 a.m. In normal condition, gene A may peak at 9, gene B might peak at noon, gene C might...
We have morning genes, we have midday genes, we have evening genes, we have night genes.
So they all have to peak at the respective time. And for a long time, we thought that you have to actually take blood samples in every 2 hours, 3 hours, 4 hours. But what you found in one of the seminal papers is you can just take one sample. The morning gene should be up, the noon gene should be slightly rising, a little below, and then the afternoon gene,
they are low, and the night genes are also in between. So every time point has a distinct, I would say, a signature or pattern. So if I take a sample in the morning, indeed, I expect the morning genes to be high and the evening genes to be low. If that is not the case, even though I took the sample in the morning, I can conclude this person may have internally still night. So, for example, a teenager, which I wake up at 7 o'clock, internally it may be for him or her 4 o'clock or 3 o'clock. And yet we send them to school and let them write exams. This is unfair because we are genetically different.
No, that's great, because so let's come back to that discovery and then this diagnostic tool. So how did you test this idea? How many patients? And then what is the current state of this diagnostic? Can people go order the test and figure out that?
So we started with a blood test. And first of all, we needed to learn more about all these morning and nighttime and evening genes.
Yeah.
So we did basically 2 studies for doing the test. The first study created a so-called training data set, where we indeed took serial samples every 3 hours in a very, very standardized cohort in a protocol which is called constant routine in the field. People do not sleep because we do not want an influence of sleep. People eat every hour an isocaloric snack, so the same snack, because we don't want to have meals to influence it, and so on. So in this first study, we got the baseline, and then asked the computer, please select the genes which are the most informative for this signature, for this pattern. And then we did a validation study, because you never do create a test and then say, okay, this is internally consistent. You do a completely different study with people from the street, not in a very standardized condition, but just taking them from the street. So first study was only 12 people because it was very standardized and very expensive. And the second study was, we had 28 people with also not so many, but this time males and females, and we always compared our new diagnostics with this gold standard melatonin test to see, is it as good as the melatonin test, or is it different? And this test is about half an hour away from the melatonin, while one should say melatonin has also a certain error. So we cannot say, so it's probably the same quality
as good as melatonin.
And then that was the first step, and we published that, and people were really interested, and then and for science it's really good, because when you do research, patients coming to the clinic and so on, and you can easily draw blood, but consumers cannot do that at home. And this is what motivated us to do another and develop another test, which is based on hair roots. So people kind of can plug out their hairs, and it's not the hair himself, but it's the little white stuff which is attached at the hair, because they are living cells, and we can measure gene expression in these cells. And so when you want to determine your own chronotype, now you can go to you can order online this test, and then you're being shipped a little test tube and an instruction, and then you plug out a couple of hairs, put it into the solution which stabilizes the cells, or the RNA in the cells, and then you ship it back to the lab, and a few weeks later you get your chronotype, and also an instruction of how to use it for your normal life, when to eat and when to sleep and when to do exercise and so on. And we have done that now with, we have about 5,000 people tested, and what is interesting, you can really see this nice normal distribution of chronotypes. You can also see how that depends on season, when people so meaning that, as I said at the beginning, this is not a chronotype fixed in stone. It depends a little bit on what you do and how much light you get. And there's a famous study from Ken Wright about graduate students going camping and kind of shifting their chronotype to earlier times.
So this test, again, I'm coming back to this test again. So when you send this kit, people can take the test any time of the day or night or evening. Is that right?
In conceptually, yes, but we have carefully analyzed all the data, and we in our recommendation is doing it in the morning.
Any fixed
time? Because it's the time when certain genes have the rise or the going down the steepest, and this makes the test at that time of the days is particularly accurate. Although conceptually it could be in principle done at any time.
I see. And then you know the And people have to mark what time they...
This is essential. We have to know from what time of the day the sample is coming.
Yeah.
So when people do it at 11 a.m., they could just write on the on the test tube 11 a.m.
And you also have the address, so from there you know what time of the relative sunrise, sunset.
Yeah, we have the address and the zip code, and we also would know whether they're living in a big city or whether they're living on the countryside, which may also be interesting to see when you assume that people living in an urban environment usually get less light than people on the countryside. And when you then compare 1,000 people from the urban zip codes versus 1,000 from the countryside, you can see that the countryside people are a little earlier.
Yeah.
Probably not because their genes are different from the urban, but the environment is different.
But can you see amplitude?
Not yet with this test, but I should say we are and this is unpublished. What we do is we have 3 different computer algorithms to predict the phase. And in most of the cases, they more or less completely agree and say, okay, 9 p.m., 8:50 p.m., 9:10 p.m.
Wow.
So the standard deviation is low. However, as you can imagine, if you ask a computer to predict a phase from a low amplitude rhythm, it makes much more errors.
I see.
Because it's not so defined.
Yeah.
So since we have 3 algorithms, the standard deviation, or how much they agree, is a measure for, or maybe a measure for, amplitude.
I see.
And we have done that for a few people where we have time series and have really the amplitude. And indeed, the standard deviation, or the agreement between the 3 models kind of predict the amplitude. But since we have no gold standard for amplitude, we have not done a validation study for this particular question.
I'm curious a little bit because recently I read an article about circadian rhythm in cats, house cats. And what I learned is house cats have very low amplitude circadian activity rhythm in urban areas, but in rural areas where they can go outdoor, they actually have a strong circadian rhythm. So
it's probably because of the feeding, the food supply in the urban areas. They have probably constant feeding, and in the rural areas they have to be active to find food.
Sort of interesting. But coming back to this, so this test, what is the name of the company, what is the test, and where this test is available, in which countries?
The name of the test is Body Clock Technologies, and it's currently available in Germany, Switzerland, Austria, but we are also thinking about expanding it to the US.
Okay.
Yeah, and then we have occasionally received samples from the US. You can find it online, Body Clock.
Yeah. And how much does it cost in
euros? I think in dollars it's about $180. Yeah. And you get not only your chronotype, you get I think a 50 pages recommendation and explanation of all the studies of when you should eat and when you should exercise and so on, and what it means and how to prioritize when you want to kind of change your life, what is best to do first. Because usually if you get a new lifestyle recommendation, you cannot do all at the same time. And this is how what you get back.
And if the same person, there might be some people who have come back for their body clock. How much is test to test variation? Of course, if they have changed their lifestyle, they would have changed body clock.
I have done it with a few people, including myself. every week
Yeah.
for a whole year.
You are a clockwork, so
Yeah, every
you
week.
must be
So
you must be the positive
it is
control.
it is pretty stable from week to week, but in the long run,
Yeah.
I see myself being earlier in the summer,
Yeah.
about an hour,
Yeah.
than in the winter. So it depends on what the distance between the tests is.
I see.
But if we have the same sample taken at the same day, but just different hair, then it's pretty stable.
Coming back to the hair, it's not only the head hair. It can be.
Or
do you prefer head hair?
Yeah, we prefer head hair because we have not systematically tested beard or eyebrows or something like this. I think to be.
So right now it's only for people who have head hair.
Yes.
Sorry.
Yeah, you have nice
I do
head
have,
hair.
but there are a lot of people who don't have.
You are.
And those,
I mean, they often they just shave them, and but as we don't need hair itself, we only need the hair roots. Then you just wait a few days and just take the forceps and pull it out.
I see. Just one hair follicle is enough, or 2 or 3 is better?
I mean, we usually, it depends a little bit on the type of hair and on the thickness. If we really have thick hair follicles, then 3 is enough. If some females with thin hair have very small ones, then we need a few more. It's all described in the instructions.
Okay.
But
so I should order. Can I order from US?
Yes, you can order. Yeah.
Okay.
Yeah.
Yeah, because I'm now curious for many of our clinical studies to see whether we can do this test.
Yeah.
So is there a difference between, say, male and female of the same age, or are you seeing those differences in postmenopausal women, premenopausal women?
We have not asked for so there is a systematic difference. Males are slightly later, about 20 minutes in a cross and corrected for age but we have not looked for systematic differences between postmenopausal and premenopausal women.
Yeah. And have you seen any surprises? Like, for example, people are sending 11 a.m. sample. Are you seeing some of them maybe even 4 p.m. that body clock, or 4 a.m.
Sometimes it's a surprise, and one surprise also was because we had an expectation from questionnaire data that with increasing age you get earlier and earlier, and we found that this is true. But when and at the very end, after 60, people get later again in our test
after
they retire. And then we ask, maybe it depends on work. And the surprise was this plasticity. So we see a nice correlation between the number of work days and the chronotype. So the less you work, the later you are, and we interpret that as people who work usually set an alarm clock and then get exposed to light much earlier, even though their internal clock would say, okay, I would like to sleep longer. But then if you get every day a morning light, then you kind of can get earlier. But if you retire, then you can live with your clock, and then the population of retired people gets a little bit later again. That was a surprise to us.
No, there must be so many inside, because truly now you have the largest data set, molecular data set of internal clock time of people. There is no one else. Even if you combine all the dim light melatonin,
there's a paper where it's about 100 or something, or maybe 150.
And you have 5,000, and it's growing.
It's growing.
So this will be really exciting. So what is next? What you foresee this test being used for in?
Okay, so there's two what is next things. what kind of test we need in the
Yeah.
future. Is it only phase? And we talked about amplitude, and what can we use with this test?
Yeah.
And to the second point, circadian medicine is that we want to, for example, use medication at the right time of day. And the right time of day is not what is on the wristwatch. The right time of day is what is in your internal time.
Yeah.
So we need to design studies for medications where we already have good evidence that time of day plays a role, and do then a precision medicine trial, or a personalized medicine trial where the internal time of the patients is taking into account. So we can either design the trials in such a way that we say, okay, we test different times in relation to internal time, or we use the test to stratify the cohorts, or we use the test for later analysis to see whether chronotype made an efficacy difference.
Yeah.
So that's when is the best time to take my medicine question.
Yeah.
And of course, there's certain pathologies where you can also use it. For example, there are certain blind people, they suffer from a syndrome called non-24.
Yeah.
So they cannot synchronize to the sun because their eyes are degenerated. So what they need to take is darkness in a pill, so some kind of melatonin.
Yeah.
But when they take it is unclear. So we would use this test to first define their phase. Where are they? Because they're not synchronized to the outside world. They might be anywhere in terms of internal time.
Yeah.
And if we use that test to define it, then we can prescribe the melatonin much more precisely.
Yeah, I had never thought about the non-24 hours and them using this test for melatonin prescription, because for them it's very tough to figure out when to take melatonin and when to. Many of them cannot keep a work for a long time because of this sleep issue.
Yeah.
That's their everyday life. But then, in terms of treating disease now, cancer is a big thing.
Yeah.
And many of the cancer drugs are not 100% effective. So what do you foresee? What is the improvement in efficacy of different cancer drugs by figuring out the internal timing?
Yeah. So the question is, how much would or in science terms, what is the effect size of personalization? This is a big question. And it's a very good question because so the short answer is we don't know. We have to do the studies to see whether, because when we talk about chronotherapy or thinking about timing the medication, then we have two levels. One is we can just say, okay, what we know from preliminary clinical studies or from mouse experiments, let's do it in the morning, because, for example, checkpoint inhibitors in cancer, in all kinds of cancers, seem to be, at least in retrospective studies, more effective when you take it in the morning. But the morning might be different. So so you could either say, okay, let's do for all the cancer patient in the morning, and then we are already on a very good track. And whether the statistics tell us this is probably a significant effect. How much we can improve it by making the morning adjusted to the individual internal morning, it's not so clear. We don't know that. We have no data about it. But I think if it's a very common disease and a very common prescription, even a small effect, if 100,000 people do it, then if we save 500 more lives, also, I mean, every life counts.
No, I think it's a chicken and egg story, because until now, the only time we could measure for drug timing was the wall clock time. We
Yeah.
did not have the body clock time measure. So I guess in future, if there is a trial where the body clock time is also measured, then I think we'll know the effect size.
Yeah, exactly.
Yeah.
So
are you collaborating with anyone who is doing cancer trial to?
Not for cancer. We do we collaborate for vaccination efficacy. We collaborate for multiple sclerosis. We have certain types of depression. So we are just in Berlin. We're just kind of setting up a huge circadian medicine consortium where we want to look at several hopefully distinct pathologies to get a kind of transdisciplinary view on circadian medicine and to identify overarching principles of circadian disruption and on circadian medicine approaches, and deliberately looking at several different pathology.
Yeah.
For example, it's interesting to see in the ICU where people are almost living in constant light.
Yeah.
That if you have light screens which are particularly designed to improve the circadian rhythms, that you the degree of delirium, which is a common consequence of anesthesia or being in the ICU, is much reduced.
Yeah. So have you done this body clock timing on ICU patients to see?
Yes, we have done that with the blood test, and and this is interesting because here you almost so the more severe the sepsis, for example, is, the less rhythmic the people are. So you cannot define a phase here.
Yeah.
You see, and if we do even serious samples, because ICU people are, you can draw blood every 3 hours because they have anyway an access. You don't see rhythms, and if you target the clock, and we call it targeting the clock by improving the amplitude, then you see it goes up again.
So what do you mean by targeting the clock?
So my take on circadian medicine is that we have three main components.
Yeah.
One is detecting the clock, which is a body clock test or maybe an amplitude test in the future. This is one important thing. Exploiting or harnessing the clock is the clock may be fine, but we just need to find the right time for the medicine.
Yeah.
And targeting the clock is not fine, it's kind of broken, and we need to strengthen the rhythm. And we can do TRE or time restricted eating to do this. We can do light to do this. We can use melatonin to do this. We can try to drug the clock or to find small molecules to do this. We can do exercise to do this. This is my conceptual view of circadian medicine: detecting, exploiting, targeting.
Yeah. No, that's beautiful. That makes it very simple and tangible for people to understand. When it comes to targeting the clock in ICU, if there is simulation of light and darkness, do you see the clock coming back in your body clock test? Have you?
We are not that far yet. Okay. Not done yet. I mean, you can use light, and another thing is, of course, feeding, because many people who receive enteral feeding receive it constantly, and we need to do it rhythmically. But my expectation or my prediction is that we will see coming it back. Even at the molecular level.
No, I think the ICU patients is very important because I think in the US alone, the number of patients who get diagnosed with the ICD code, sepsis, is almost as many as those who are getting a cancer diagnosis. And yet, the treatment in cancer, it's very well defined, different types of cancer, different grades of cancers, and then treatment is given. But sepsis puts a lot of challenge because everything has to be diagnosed within hours. There is not even a day. And in those cases, for example, if we target the clock, just like you said, let's give drug in the morning, checkpoint inhibitor for everybody, and then a lot of people may benefit, and then personalize. Similarly, in case of sepsis or in case of ICU in general, if we come up, of course, that has to be done with some very specific clinical trials or studies that this type of enteric feeding, where instead of continuous feeding, 12 hours feeding or 10 hours feeding actually improves the clock, irrespective of how severe the patient is. That can be the first step.
Exactly, I couldn't more agree.
And you are at the biggest hospital, university hospital, so I guess you are the right place at the right time with the right expertise to do that.
Yeah, we will do this.
Yeah. What else do you see as the future of circadian medicine, and what are the barriers?
Yeah, I mean, the barriers are the daily routine in the clinics. I mean, I have several colleagues who said we have been changing our routines very often. We have adapted to the pandemic, and we had logistic routines, wearing masks, and kind of all the visitors, and that. So we can do this. And if you give me the evidence, so what we need as our field, as chronobiologists, circadian medicines, we need more randomized controlled trials. We need more. We need meta-analysis. We need to know the effect sizes to convince our clinic directors to really implement it. Because if we say, okay, for cancer immunotherapy, this morning treatment is compatible with the daily routine. But if we say, okay, heart valve operations should be done in the night, this may be a little bit problematic. And also because it's always also about money and kind of make optimal use of the operation rooms, and you want to do it all day long. And if you say no, in the afternoon you shouldn't do it, then they're empty. So we need to reorganize it, because some operations may be beneficially to be done in the afternoon and so on. So there's practical and logistic barriers, and there are scientific barriers, because we need better data, and we need really to get these data or get this insight into the guidelines, into the recommendations, and this is a long way. Although it may not be as long as for getting a drug on the market, because we have already our circadian medicine approaches, and our circadian medicine approaches can go right away into the clinic, because they're either approved. So we have the drugs, and the timing is free usually for the doctors, and our targeting the clock approaches, light and melatonin and feeding, these are all safe. These are safe interventions. So we are much better off than a classical small molecule, which is a new drug. So that will save us a lot of time, but we have to do these studies.
But let's come back to one simple thing that can be done. For example, in this conference, and there are many papers showing that many types of vaccinations are more effective if they're given in the morning. Can that be put into practice in a university hospital like Charité?
Yes, I mean, if we have the next pandemic, and we have a very good colleague who is a vaccination expert, who is just doing a study together with Christoph Scheiermann, who has very nicely published that anti-cancer vaccination is highly time of day dependent, and which is a little bit, at the first glance, counterintuitive, because your vaccine depot is there for several
Many
days. And why should the time of but apparently the dendritic cells, the first encounter of the dendritic cells, make the difference. And yes, I mean, we will do these studies, or they will do these studies, and I will watching.
Because that can be,
advising, yes.
you know, if you go back to the diagnosis for diabetes, it became very clear that having a fasting blood glucose, typically in the morning, because people can overnight fast, is much better than any random blood glucose. So that became the standard of practice now, that all over the world, doctors ask patients, okay, so come back to clinic, fasted in the morning, and we'll take your blood. So similarly, I think there is an opportunity that within 5 to 10 years, there should be a standard recommendation, not essentially a strict guideline, that you should come get your vaccine before noon.
But imagine during the pandemic, when we had to vaccine several hundred million people at a very short time. Then we come back to these practical things, and being vaccinated in the afternoon is better than being not vaccinated.
Yeah, of course.
So that's right. So if we have a huge wave of vaccination need, then this may be not doable.
No, I'm saying the regular vaccination, where people have to go get, like for example, older people, they're going to get their shingles vaccine, and
or the influenza vaccine.
Influenza vaccine. So those things which can be scheduled.
Totally agree.
They can be. So I guess we have to figure out what more evidence is needed to nudge the healthcare system to make that recommendation or guideline or suggestion when they're asking people to come sign up for vaccination.
I think we need I think we need to come from both sides. We need to do multi-site trials, randomized, placebo-controlled. Well, maybe not placebo-controlled in this case. And we would also better have in mechanistic insights, similar what Christoph is doing. And in explaining why that morning vaccination is so much better than the afternoon vaccination.
No, actually, we had Christoph yesterday in the last episode where he talked about this in great length, and he also brought up the barriers and what is needed to be done. And this is really exciting to see how much is going on in circadian immunology. Just imagine 15 years ago when you published that macrophage circadian oscillation.
Well, how much has been achieved? Now we have immune anti-cancer vaccination, which is more effective at this time of day. So we didn't have both. We didn't have anti-cancer vaccination, and we didn't have time of day. Now we have both, and the next step is to personalize.
So super excited. Thank you, Achim. And I hope to see next time there will be maybe 50,000 or 500,000 data. And then that's the time that then we'll have a lot of fun to figure out how this molecular marker of body clock changes through development or through age. Like you said, how retirement changes it, or how night shift work, how day shift work, or time restricting of drugs, all of this change. So that's super excited.
Thanks, Satchin, for having me.
Thank you.